Imagine you wake up one morning and discover your body is running on buggy software. Not, "I forgot my password" buggy. I mean the original source code, the DNA code, has a typo so serious it breaks your blood, your muscles, your vision, your whole life. Now, here's the plot twist. In the last few years, doctors have started doing something that sounds illegal in a science fiction movie. They're not just treating symptoms, they're delivering a new genetic instruction, like shipping a patch update into your cells. And this isn't a one-day story, this is a right-now story. The US FDA lists multiple approved cellular and gene therapy products on its official approvals page, updated through December 9th, 2025. So today, let's tell the true story of gene therapy. With heroes, disasters, plot twists, ethical dragons, and the strange truth that a virus can be turned into a medicine delivery truck. And yes, there will be humor. Because if we can't laugh while discussing customized viruses, what are we even doing? Smoke versus fire. Most medicine, historically, is smoke control. You've got inflammation, reduce inflammation. You've got pain, reduce pain. You've got low levels of something, replace it. That's not bad, that's life-saving. But for many genetic diseases, the symptoms are the smoke. The fire is the DNA instruction itself. Gene therapy is the radical idea that instead of fighting smoke forever, you walk into the building, find the faulty wire, and carefully replace it. Trail marker number 1. DNA equals the instruction manual. Many diseases equal a single-letter typo with massive consequences. Gene therapy equals deliver a correct instruction or edit the wrong one. And if you're thinking, wait, are we doing Windows updates on humans? Yes. Except if your laptop crashes, you lose a spreadsheet. If your liver crashes, you lose the ability to continue being a person. Here's the problem gene therapy had to solve. Even if you have the perfect genetic patch, how do you deliver it? Cells don't accept random USB drives. So scientists looked at nature's greatest delivery expert, viruses. Viruses are basically biological FedEx, except the package is usually make more virus. Gene therapy takes that delivery truck, empties the harmful cargo, and loads it with something new, therapeutic genetic instructions. A major workhorse is AAV, adeno-associated virus. It can deliver genetic payloads effectively in many tissues, but it's not magic. It has dosing limits, safety limits, especially at very high systemic doses. Trail marker number 2. The therapy is often the gene or genetic editor. The vehicle is a vector, often a virus-like delivery system. Engineering a safe delivery system is half the battle. It's like hiring a former cat burglar to install security systems. You don't trust him because he's innocent. You trust him because he knows every door. Every hero's journey has an abyss. Gene therapy's abyss has a name: Jesse Gelsinger. In 1999, he joined a gene therapy trial for a metabolic disorder. He received a dose using an adenoviral vector and suffered a catastrophic immune reaction. He died days later. That moment hit the field like a meteor, and it forced a hard truth into the open. Delivery is not a detail. Delivery is the danger. Then came another warning. In early SCID gene therapy trials, some patients developed leukemia. Cause? Insertional mutagenesis. Viral vectors integrated near genes controlling cell growth, pushing cells toward cancer. Trail marker number 3, very important. two major historical risks emerged. One, immune overreaction to vectors. This can be fatal. Two, insertional oncogenesis, vector integration triggering cancer. Result? Gene therapy didn't fail, it got disciplined. Science is the only profession where you can say, "Good news, we learned a lot." And what you mean is, something went horribly wrong and we will never forget it. A comeback story needs a symbol. For gene therapy, one defining symbol is Luxturna, a therapy for a rare inherited retinal disease caused by mutations in RPE65. In December 2017, Luxturna became the first FDA-approved gene therapy in the U.S. for an inherited single gene disease. This was a turning point, not because it was the first idea, because it was a real therapy with real regulatory approval. And it showed the modern recipe. Choose a disease where the target tissue is reachable. The eye is ideal for localized delivery. Deliver a functional gene copy. Measure real-world function. And follow patients long-term. Trail marker number 4. Gene therapy moved from promise to products. Regulatory approval signaled this is medicine now. Luxturna was basically science saying, okay, we're not gods, we're not wizards, but we can fix one page of the manual in one organ, carefully, with supervision and a mountain of paperwork. Now we need the map. Modern gene therapy mostly comes in two flavors. In vivo, treat inside the body. You inject the therapy and it travels to target cells. Ex vivo, treat outside the body. You remove cells, modify them in a lab, then return them. Ex vivo is like, I don't trust the neighborhood. Bring the patient's cells into the workshop. This matters because ex vivo can reduce some delivery uncertainties, but may require intense preparation, like chemotherapy conditioning for stem cell therapies. And today, this isn't theory. The FDA's approved list includes multiple gene and cell-based gene therapies, including Zolgensma for spinal muscular atrophy, Hemgenix for hemophilia B, Roctavian for hemophilia A, Casgevy, a CRISPR-edited therapy for sickle cell disease, Lyfgenia, another gene therapy for sickle cell disease, and several others, updated through December 2025. Trail marker number 5. In vivo equals direct delivery. Ex vivo equals modify cells in a lab, then return them. Multiple FDA-licensed gene therapies exist today. In vivo is like DoorDash, efficient, but you pray the driver doesn't get lost. Ex vivo is like, no, I'm picking it up myself from the kitchen while staring the chef in the eyes. Boss number 1, safety. Safety isn't optional, it's the whole game. Because gene therapies can have long-lasting effects, regulators emphasize long-term monitoring. The FDA often recommends follow-up for delayed adverse events, commonly up to 15 years. And real-world safety concerns still appear. For example, in late 2025, reports noted the FDA added a boxed warning to Sarepta's Elevidys after fatal liver injuries in certain pediatric patients. Use was narrowed, monitoring increased. That's not a gene therapy is bad story. That's a this is powerful biology story. Boss number 2, cost and access. Many gene therapies are one-time purchases with extremely high prices. Reasons include small populations, complex manufacturing, uncertain long-term durability. Luxturna famously sparked pricing debates and discussions about outcomes-based payment models. Boss number 3, the forever question. Does it last 5 years? 10? A lifetime? Sometimes yes, sometimes no, sometimes we're still watching. Trail marker number 6. Long-term follow-up matters. Safety signals can change labels and eligibility. Access remains one of the hardest non-science problems. Gene therapy is the only kind of one-time purchase where the warranty is 15 years of checkups. Classic gene therapy often adds a functional gene copy, but now comes the newer twist, editing the DNA itself. In December 2023, the FDA-approved Casgevy, the first FDA-approved CRISPR-Cas9 gene editing therapy for sickle cell disease in patients 12 and older.
Before mass printing, knowledge spread by copying. Slow, error-prone, controlled. Then came the printing press. Information could replicate faster than power could contain it. Gene therapy is the printing press moment for biology. Not because it makes humans perfect, because it makes biological instructions modifiable. But every printing press era has propaganda, counterfeit pages, censorship battles, and people screaming, "This will destroy society." So, here's the rule. The deeper the power, the deeper the responsibility. That's why modern gene therapy is conservative where it matters. Controlled manufacturing, trial phases, long-term monitoring, intense regulation. Let's clean the fog. Gene therapy is real, approved, used in multiple serious conditions, often aimed at rare genetic diseases, blood disorders, and some cancers. Sometimes a one-time treatment. Gene therapy is not a casual upgrade you get at the mall, a DIY biohacking project. Please don't. A guaranteed permanent cure for every condition, because it's still powerful biology. Immune responses can be dangerous. Some delivery methods carry toxicity risks. Long-term unknowns require long-term follow-up. If somebody online says, "I can do gene therapy in my garage," that is not a startup. That is a horror movie. So, what do we return with? A new way to think. For the first time in history, humans can treat disease by changing the instructions inside living cells. And regulators now list multiple approved products because this isn't theory anymore. But the hero of this story is not a scientist alone. It's the system, the patients who took the risk, the clinicians who monitored outcomes, the regulators who demanded evidence, and the researchers who learned from every failure. Gene therapy didn't become real because humans became arrogant. It became real because biology forced us to become humble. Final trail marker. Gene therapy is not "we control life." It's "we've learned to negotiate with it carefully." If you want part 2, we can go even deeper. Why some therapies target the liver, why the immune system is the real final boss, how CRISPR differs from base editing and prime editing, and what it would take for gene therapy to become affordable worldwide. Drop a comment. Would you take a one-time gene therapy if it had a small risk but huge upside? And tell me which disease area you want next: blood, muscle, eyes, or cancer. Subscribe, because the code patch era has started.
The simulations confirm our hypothesis.
And the samples show the same pattern.
Excellent. Let's proceed to the next phase.
The results are in. They're very promising.
That's excellent news.
This could be a breakthrough.
Let's review.